Creatine, widely known for its role in enhancing muscle performance, is beginning to garner attention in an entirely different and far more urgent domain: the fight against Alzheimer's disease. While its reputation as a sports supplement has made it a mainstay in athletic circles, a growing body of scientific interest is exploring how creatine might support brain energy metabolism, particularly in neurodegenerative diseases where metabolic failure is a key feature. In Alzheimer's, where brain cells struggle to produce sufficient energy, early research is pointing to creatine as a possible means of preserving cognitive function and slowing disease progression. Although still in the early stages, this line of investigation represents a potentially transformative shift in how we approach neuroprotection and brain health. Alzheimer's disease is marked not only by the accumulation of amyloid plaques and tau tangles, but also by a profound breakdown in mitochondrial function. Mitochondria, the energy-producing organelles of cells, are impaired in Alzheimer's, leading to reduced production of ADP, the molecule that fuels virtually every biological process, including synaptic transmission and memory encoding. When brain cells become starved for energy, they can no longer maintain the complex electrochemical activities needed for cognition, resulting in the characteristic decline in memory, reasoning, and behavior. Creatine's ability to replenish ADP through the phosphocreatine energy shuttle makes it a compelling candidate for intervention. In this system, creatine acts as a phosphate reservoir, rapidly regenerating ADP in high-demand situations. This mechanism, well understood in muscle tissue, is increasingly being recognized in the brain, where neuronal activity depends heavily on a stable energy supply. In individuals with Alzheimer's, where ADP levels are chronically depleted, creatine could theoretically serve as a buffer, supporting cellular energy demands and improving neuronal survival in metabolically stressed regions of the brain. Preclinical studies in animal models of Alzheimer's have shown some encouraging results. Mice given creatine supplementation exhibited better memory performance, reduced oxidative stress, and greater preservation of synaptic function compared to control groups. These findings are supported by observations of higher mitochondrial integrity and fewer markers of inflammation in the brains of creatine-treated animals. These effects are particularly notable in the hippocampus, the region most critically involved in memory and most vulnerable to early damage in Alzheimer's disease. While human studies remain limited, preliminary clinical trials and observational data suggest a potential therapeutic value worth exploring. Creatine supplementation has been shown to improve aspects of cognitive function in older adults without dementia, and its neuroprotective properties are being evaluated in ongoing trials involving mild cognitive impairment, MCI, a condition often preceding Alzheimer's. Moreover, creatine's favorable safety profile and low cost make it an appealing candidate for adjunctive therapy, particularly in the early stages of cognitive decline when interventions may be most effective. However, it's important to be cautious. While the biological rationale for using creatine in Alzheimer's is strong, the current evidence is far from conclusive. Much of the supportive data comes from animal studies or small-scale human trials, and there is no definitive proof yet that creatine can alter the course of Alzheimer's in a meaningful way. Large, randomized controlled trials are urgently needed to determine whether the benefits observed in experimental settings translate to clinically significant outcomes in human patients. Additionally, the optimal dosing strategy, duration of use, and potential long-term effects in elderly or frail populations remain uncertain. There is also the concern that interest in creatine may outpace the science, leading to premature use by patients or caregivers desperate for solutions. As with any supplement, especially one used in vulnerable populations, self-prescribing creatine without medical guidance could introduce risks, especially if taken alongside other medications or in the presence of underlying kidney disease, which is more common in older adults. While creatine is generally well tolerated, the need for medical oversight is particularly important in the context of chronic neurodegenerative illness. That said, the broader implications of this research are undeniably exciting. Creatine represents a shift in how we think about Alzheimer's, from a disease of protein misfolding to one of failing energy metabolism. This metabolic perspective opens the door to new strategies that focus not just on clearing plaques, but on fortifying neurons against the metabolic collapse that precedes cognitive decline. It aligns with a growing interest in mitochondrial therapeutics and may ultimately integrate into a multi-pronged approach that combines lifestyle interventions, medication, and targeted supplements. In conclusion, creatine is emerging as more than just a performance enhancer. It may also be a neuroprotective agent with potential applications in the early stages of Alzheimer's disease. While definitive answers are still years away, the current findings provide a strong foundation for continued research and clinical exploration. If future studies confirm its benefits, creatine could become an important part of an integrative strategy to maintain brain energy, support cognition, and delay neurodegeneration in one of the most challenging diseases of our time. Until then, its promise should be tempered with prudence and scientific rigor.